Can you see it now?
Yeah, we are able to see the. Yeah, it's good to see that. Yeah, good to go. I think we will start the session by around 4:50 p.m. 4:55.
Okay, sounds good. I'll get ready. I'll be in the office room by then. I'm just sitting in the couch right now. But,
yeah, fine.
All right, sounds good.
Yeah, we'll ping you.
All right, thank you.
Yeah, thank you. Yeah.
Bye.
Hi, can you hear me? Hello, can you hear me? Hello? Hello?
Hello.
Hi, Suma, can you hear me?
Hello.
Hi, can you hear me?
Yeah, I can hear you, Harsha.
Okay.
Shall we start after another 5 minutes? 5-10
Yes,
minutes.
yes. It looks like there's 4 minutes from to 5:30 p.m. But
Yeah,
yeah,
we'll start at sharp 5.
okay, sounds good.
Yeah.
I'll be ready.
Yeah.
Yes.
Dear participants, we would like to welcome you all to today's session, webinar on genome engineering. Today we are having Dr. Harsha K Rajasimha here with us. Before starting the session, would like to say a few words about Dr. Harsha K Rajasimha. Dr. Harsha K Rajasimha is a founder and chairman of Jeeva Informatics Solutions Inc., located at Vienna, USA. He is also a founder and chairman of Indo-US Organization for Rare Diseases. So it's a quote from an Aristotle. As per Aristotle, he quotes, "The ideal man bears the incidents of life with dignity and grace, making the best of circumstances." So one such circumstances has made Dr. Harsha to come up with a research on rare diseases. After losing a child to a rare congenital disease in 2012, Harsha got determined to apply his years of postdoctoral clinical genomics data research experience at National Institutes of Health and Industry Product Development experience to develop products and solutions to help accelerate clinical research. He is a social entrepreneur, is committed to apply his 15 plus years of distinguished experience in healthcare and life sciences, consulting, precision medicine, genomics of rare diseases and cancer to help accelerate drugs, R&D, and digital therapeutics. As a founder and CEO of Jeeva Informatics, he is on a mission to bring fresh perspectives and innovations to bring faster cures by engaging patients as participants in clinical research. As a software architect, he has led the development of some of the largest interdisciplinary big data projects in bioinformatics, including Pathogen Portal, Pathosystems Resource Integration Center, which is funded by NIH, Cancer Biomedical Informatics GRID, it's a CBIG funded by NCI, Cyber Infrastructure Development for National Eye Institute's Genomic Program funded by NEI, Laboratory Information Management System implementation at the FDA Office of Regulatory Affairs and Genome Explorer, an enterprise integrated platform for next generation sequencing data analysis. His contributions to the genomics program at NEI has earned the Director's Innovation Award in 2012. As an entrepreneur, he founded Jeeva Informatics Solutions in 2013. Jeeva is a life sciences big data informatics company which has received the Bioscience Company of the Year award. In addition, he co-founded the non-profit organization for Rare Diseases India and served as a board member. He has led strategic business development initiatives while simultaneously delivering in technical and scientific roles. In collaboration with the Rare Genomics Institute, he received the Sanofi Genzyme's Rare Diseases PAL Award in 2016. As a scientist, he has authorized 15 plus peer-reviewed articles and book chapters and participated in 30 plus talks, events, and conferences as a keynote speaker, delegate, invited vendor, and our host. We welcome you, Dr. Harsha K Rajasimha, for today's session. Over to you, Dr. Harsha.
Thank you so much for having me, and thank you for that very kind introduction, Dr. Sumalata. Just wanted to add that I went to college in Bangalore University with Sumalata about 20 years ago when we graduated together. So it's a pleasure and honor to be back on a webinar with you.
Pleasure to have you, Dr. Harsha, here. Yes. Could you please share the screen, Harsha? So let's go. Yes.
Can you see my screen now?
Yes, Dr. Harsha.
Okay, thank you. Yeah, so what I will do today is walk you through my 20 years of journey since graduating with a Bachelor of Engineering in Computer Science and Engineering from Dr. Ambedkar Institute of Technology in Bangalore in 2000. And since then, I have taken probably an unconventional and an unusual career path, which might be of interest to many of the engineering students that may be thinking of various options, and provide some career guidance or coaching to some of the engineering students right now, as well as reflect on my thoughts on how engineering education may have evolved over the years, and from an industry perspective, how engineering education could potentially consider evolving to make sure these graduates will be more readily useful for the industry. So from software engineer to social entrepreneur, my journey has started from Dr. AIT, and I came to Virginia Tech in Blacksburg, Virginia for a master's in computer science. And I did not have plans to do a PhD, but I got interested in bioinformatics and I went on to do a PhD as well. I'll share very little highlight on what I did for my master's thesis in computer science at Virginia Tech. I developed a methodology called PathMeld for the unification of metabolic pathway databases. What that means is these, this is the traditional way of integrating databases. This was about 18 years ago. So there are better ways to do things now. But back then, what we did is you took two of the metabolic pathway databases, KEG, which is a Kyoto Encyclopedia for Genes and Genomes from Japan, and Metapsych, a similar metabolic pathway databases curated in the United States by SRI International. Since they were both supposed to have this content related to pathways, but curated in two different countries, completely different teams, we wanted to see what useful information we can glean by combining information from both those sources. So our approach was to download the flat files, the source flat files, reverse engineer them, engineer the source database schema, and transform those schemas into a common unified schema, and use that to then load the database with the combined information from both the databases. There's a good bit of work after that to establish entity correspondences between the two database sources. So may seem familiar for most um, folks in the computer science and information technology space. The complexity lies in the domain of how complex these metabolic pathway genome databases are and how complex the relationships between them could be, as well as the naming conventions. If you remember high school biology or chemistry, you may remember names of chemical entities like 1, 2, 3 triphospholochlorine or something like that, right? So, and there are different ways of writing those chemical formula, and so to do string matching, exact matching versus approximate matching to combine information from different databases was not trivial. In any case, the unification schema that I came up with for my thesis pretty much captured the essence of relationship between a chemical reaction or a biochemical reaction, which is what makes a metabolic pathway, and one or more reactions, right? So it's got to be two or more to call it a pathway. And these metabolic reactions are catalyzed by enzymes, which are a type of protein. And these proteins are encoded by genes. And they have either one to many, many to many, or many to one type relationships. And they have attributes like various attributes, which are not all comprehensively shown in this diagram. So starting with this is where my interest in the life sciences started. As I was completing this, there were major press releases back in between 2000 and 2004. There were multiple press releases coming out about having completed the Human Genome Project. So these databases were largely for bacteria, virus, including coronavirus. Coronavirus is not a new virus, by the way. It existed several decades ago. This is a novel strain of coronavirus, but the SARS coronavirus already existed several years ago. And so but the human genome was just then getting sequenced and annotated, and that was majorly all over around that time. So I got interested in exploring how this entire network might look like for humans, and how computationally we can analyze and understand the human genome. That got me interested in continuing my work in the bioinformatics space, and continued with my doctoral dissertation in insights into mitochondrial genetic and morphologic dynamics gained by stochastic simulation. This, with Dr. David Samuels being my advisor, resulted in 3 journal publications. What we did here is looked at mitochondrial DNA, which is a part of the cell in a slipper-like organelle called mitochondria, which you may remember from high school biology. So within those slipper-like structures, you have multiple DNA loops, circular loops, which are almost like a bacterial genome, but it's part of the overall human genome, but part of not in the nucleus where all the DNA and chromosomes are sitting. So these mitochondria can be in several hundreds or thousands of mitochondrial DNA in a single cell. And we developed a stochastic simulation model of how these mitochondrial DNA molecules would get distributed to daughter cells during cell division, and how the number of or proportion of mutation in the single cell gets distributed when we have the overall organism is developed after 20-30 generations of cell divisions, resulting in millions of cells, or even tens of millions of cells. So that entire simulation we were able to do in under 24 hours with our computational simulation model. And we had some pretty interesting results that were published in some of the top-notch journals like Nature and American Journal of Human Genetics. So that gives you a sense of how interdisciplinary my career, early days of my career, where I was still studying grad school, began. And since then, I have moved on, worked in various governmental labs, academic research labs, and went into industry. And I will share a little bit more glimpse into some of the work I did, and how I never dreamt, having started in a computer science engineering course, and still continue to apply the same software engineering skills. But I will hopefully be able to motivate how I'm doing the same type of work, but in a very niche life science market or industry, solving very specific problems. So along the way, like Sumalata mentioned, we had a mishap in around 2012, and that, as I was going about my day-to-day life as a scientist, and when we had a child that was born and passed away with a rare congenital disease, that got me into patient advocacy and social entrepreneurship. So I'll talk to you all about that briefly. So having started as a software engineer, I did an internship program at Microsoft back in 2002. And as I was going through my grad school at Virginia Tech, I was growing into a bioinformatician, and later analyzing big data coming from genome sequencing data sets from bacteria and virus, I had already become a genomics data scientist back then, although data scientist is more of a buzzword these days. And then when I got into the industry, I was doing business development in the space of precision medicine, which is essentially taking genomics into the clinic instead of in the lab where you're doing bench work and generating data, understanding the basic biology and medicine. You're actually now applying that for clinical practice. That's precision medicine. And I have been a healthcare and life science consultant in with the biotech pharma industry, looking at how big data and digital health technologies can help speed up the process of bringing drugs to market faster. And I have been a rare disease patient advocate, where we see that millions of patients suffering from these rare diseases receive little attention, little funding, and no public policy for a long time. And that's where I started advocating that we need that, particularly in India. In the United States, we do have a policy for Orphan Drug Act for rare diseases since 1983. United States became the first country when we got that act. However, when in 2012, 2013, when I looked at India, we did not have a national policy for rare disease. It was neglected or ignored. And I'll finally talk to you about the specific social problems that I am trying to solve in my ventures, Jeeva Informatics Solutions, and the nonprofit organizations. So I'll give you a little bit more of the science, and then continue on my journey. So the most important thing anybody needs to know about the whole space of genomics or precision medicine is the central dogma of molecular biology. For unlike computer science, which it has its roots in computation, mathematics, which has a time immemorial history, but the computer science itself is still relatively a young discipline compared to biology, which is thousands of years old. And people started observing and doing biology a long time ago at the organism level. And to get down to the organ systems level, organ level, and the tissue level, was an evolution. But these days, the genomic space is all about looking at the DNA level of individual organism. But what was believed in biology for a long time is that the DNA, which sits in the nucleus, gets transcribed into RNA or messenger RNA that travels into the cytoplasm, and that gets translated into protein. And proteins are the molecules that perform all the function in our body. And this traffic was believed to be strictly unidirectional, that it can only go in this one direction. However, there have been Nobel Prize winning inventions around reverse transcription, where messenger RNA can get reverse transcribed into complementary DNA, and not all messenger RNA that is transcribed gets translated into protein because of a mechanism called RNA interference or gene silencing. So knowing that the central dogma is no longer as valid as it was believed for maybe 100 years, because of reverse transcription, gene silencing, and many other intermediate regulatory mechanisms that have been recently discovered, is the most crucial hallmark of understanding the whole molecular biology field. Essentially, when we say omics or genomics, proteomics, metabolomics, or proteomics, right? Genomics is in the space of DNA, looking at the entire DNA in the human cell, versus transcriptomics, looking at all the messenger RNA content in the cell, an indication of which part of DNA is actually transcribed at any given time, which is primed to get translated into protein for performing a certain function. In the world of omics, you are looking at everything, all DNA, all RNA, all proteins, and that's the opportunity for computer science in biology, is to be able to digitize all the parts that make the system biology, right? So that's so I'm an affiliate faculty in the School of Systems Biology at George Mason University, where we study systems biology and systems medicine, which is basically the sum of all the parts. And these are the parts of molecular biology, the main parts. The only other part are the ligands that are missing here, which is a major aspect of metabolic pathways, like we just saw. So I'll focus. We won't have time to cover all of them, but I will describe the DNA sequencing technologies. Back in 2000 to 2007, there was a lot of sequencing platforms that were competing for the $1,000 genome prize. But now the major human genome sequencing platform has emerged to be Illumina, and they have pretty much monopolized most of the genome sequencing instrumentation market. Almost everybody uses Illumina for whole genome sequencing. So why this is so crucial is, as a computer science engineering major, I was taking pride in being part of the most fastest growing sector, right? Semiconductor industry. We know the Moore's law, and how the number of transistors in a unit area of a chip has been doubling every 2 years. What really amazed me is that the cost of genome sequencing has been decreasing at the square of Moore's law for a long time now as well. And so this is beating the semiconductor industry in how quickly genome sequencing, which cost $1 billion in 2001, to now it's under $1,000. Any of us or most of us will be able to afford getting a genome sequenced by swiping a credit card today, whereas just 20 years ago it was unimaginable, and United States government had to spend $1 billion to get that entire human genome mapped. The challenge, however, is that we now have a $1,000 genome, but a $1 million interpretation. What that means is our ability to digitize humans through genomics and genotyping depends on these 700,000 medical practitioners in the United States, and perhaps similar number in India, but only a small fraction of those physicians are able to interpret genomic data yet, from what it means to the human health. And that's where the big opportunity is, both on the medical side as well as on the engineering and data science side, to enable better and faster interpretation of genomic data. So once you have the genome sequence, you're not going to get a single sequence of the entire genome. The human genome is comprised of 22 pairs of autosomes and a pair of sex chromosomes, XX in females and XY in males. Now, what you get is a readout of the genome in pieces, which are all of equal length, say roughly around 100 to 200 bases. And if you remember the basis of a deoxyribonucleic acid, DNA, it's ACGT, adenine, guanine, thymine, and cytosine. And these reads of 100 bases or 150 or 200 bases will be a read comprising of those 4 bases, ACGT. Now, you get millions of reads in a single human genome, and you take individual pieces of these and map it against a reference genome, and these reads stack up or map against the genome at one or more places because there is gene duplication and there are pseudogenes and others, as well as these are not necessarily all unique 200 bases in the genome, as the combination, some of these sequences may be repetitive, and so you have to sort out and see where exactly uniquely they map and remove those duplicate mapping reads, and then look at which genes are where is the mutation in this particular genome sequence. So that's the computational sequence alignment problem, right? And so when you get through that, when you get into the messenger RNA and protein level, you will see that the amino acids, there are 20 of them, whereas the deoxyribonucleic acids are only 4. So you need to map from messenger RNA to protein or amino acid sequence. You need to decode the genetic code, since it's degenerate with 20 amino acids, but you have 64 combinations of the triplet codons that with 3 bases of each amino acid. And so when you get, so I'll skip through the RNA and transcriptomics, but within the genome, 98% of the genome is junk DNA. We don't understand what 98% of the genome does, even now as a scientific community. Less about 2% of the entire genome, what we call exome, is what really matters, because we understand somewhat, even those we don't fully understand. There are about 22,000 genes roughly, and out of those, about 6,000 or so genes have been implicated in human disease. And so those genes are the ones where a majority of the focus is, and new genes are being associated with the genetic disease every month. We see several publications where new genetic mutations are related to a specific disease. But to identify those genes and sequence them, we only need to pick the green boxes here, which we call exons. Those are the only boxes of the genome that get transcribed into messenger RNA and eventually translated into protein. Not the introns, they get cut out. And so we have biochemical methods to only pick the exons and sequence them, and that's both cost effective and more meaningful in the near term, based depending on the experimental need, because those are the ones that are relevant for human disease. And so most of the data you will find will be exome data and not genome data. So once you get through this biochemical process of using some sort of a magnet and pull down the green boxes that I just showed and sequence them, what you get is raw sequence data. And this is all a wet lab activity, which I never really mastered the lab side of things. My role starts as a bioinformatician once the data is generated, and taking that raw data and processing them through computational pipelines for analyzing using various existing algorithms, new algorithms, and filtering them, mapping and identifying different types of mutations. There are substitution mutations and insertions, deletions, and so on. So this was a paper we published while I was at the National Institutes of Health on the method for analyzing exome sequencing data. And the field has matured since then. There's best practices workflow that's put out by the Broad Institute of MIT and Harvard, which has one of the standard ways of analyzing genome or exome sequencing data now. where pre-processing of the data and discovering various types of variants or mutations, and then conducting analysis of what that variation or mutation means to human health. So that's to give you a glimpse of the type of analysis, the basic analysis that one can do with the data coming from genome sequencing. And you also need highly complex data visualization tools like the integrated genomics viewer as an example. You have sample 1, sample 2, 3, and 4, and you see for the same reference genome for a particular gene where you start with a few million um, single nucleotide variations or polymorphisms, and you want to identify needle in the haystack, which particular out of these 4, if 2 are, for example, disease patients and 2 are normal, which is the mutation that's only present in these 2 samples but not in the other 2, right? So that's the type of thing that you want to look for typically in a exome sequencing experiment. When you do that and apply various computational methods, you do risk the chance of accidentally losing the actual variant. But if you choose the filters carefully, you will end up with a handful of genes which you can analyze using these visualization methods and identify mutation and verify the location of that specific mutation in the genome. So what happened around the human genome is that a lot of hype, as it always happens with any technology. Genome sequencing technology was no exception. It had the hype and promise that it would help solve a lot of the human diseases fairly quickly. And it did solve several diseases, and in terms of helping identify the specific gene and mutation that's associated with a particular disease. For example, the cystic fibrosis gene was identified way before the human genome sequence was done. However, the treatment or a drug to treat cystic fibrosis did not come out until 2015 or so by Vertex Pharma. So it took about 35 years after the genetic mutation in the gene CFTR was directly associated with cystic fibrosis with very little doubt. In terms of it's a rare mutation with a very low frequency of occurrence in people, in population, and the effect size is almost close to infinity, which means it has a very large effect size. If it occurs, it's likely that a person harboring this particular mutation in a cystic CFTR gene will have the disease. But that's not true for many other genetic diseases. For example, APOE4 is a gene associated with Alzheimer's, but not everyone having the gene mutation will develop the disease, Alzheimer's disease, at old age even. So some may develop, some may not develop. So the effect size is high, but it's not close to infinity. And so it's a probability, maybe 70% chance of getting Alzheimer's, but not necessarily, and some may get at 90 years of age and not 70 years of age. So different diseases and gene mutations have different levels of frequency of occurrence. A gene mutation that's more frequent and commonly occurring in the population, like the prostate cancer gene, which has almost half of the population have that gene mutation, but the effect size is also very small, because if the effect size was high, half the population would be dead only because of this genetic mutation, given the allele frequency. But that's not the case, and so luckily the effect size is rather small. So patients who do have prostate cancer are likely to have this mutation, but not everyone who has this gene mutation will get prostate cancer in their lifetime. So that's how you interpret. So now these are the types of complex genetic mutations that still we have not been able to crack, and that's where the promise of genomic medicine is still under not out of the woods. And there are new methods and new ways of looking at multiple genes involved in a single disease right now. But most of the single gene disorders have been identified. Now the next question is, what do we do? How do we go from knowing that yes, there is a gene associated with multiple sclerosis with a fairly good effect size but not very high? How do we go from here to developing a drug or treatment for this particular disease? And that takes a long time. You have a lab with millions of dollars in funding only to study this one gene and understand the functional implications. You remember going back to the network biology or systems biology? You need to look at what are the messenger RNA and proteins that this gene transcribes and translates into, what function they perform, which organ systems this gene is expressed in, because not all genes are expressed in all organs. Some genes only have a function in the eye. They have no business to be expressed elsewhere. And some genes have a business only in the kidneys. They don't have any other business anywhere else. So the biological system has the nature takes care of itself in terms of which gene is expressed in which organ system. But where the work is going is knowing which gene is involved in a genetic disease to how do we find a treatment to now make this patient better. So, we kind of got deep into the genomics. Now, taking a step back, um, because there are many diseases where just having a genetic mutation does not guarantee the disease. Why is that the case? It could be environmental factors. So having a genetic mutation, but also being exposed to a specific environmental factor will trigger the disease potentially. Right. So, for example, for Alzheimer's, they say that keeping the mind active and being involved and doing mathematical, analytical thinking and activity can reduce the risk of Alzheimer's. So that's human behavior and lifestyle. So behavior, lifestyle plays a very important role in the health of the population. So genetic mutations can mean a guaranteed suffering in the case of many rare genetic diseases, but it's not necessarily the case for many others where behavior and lifestyle can help and do have been proven to help in overcoming the deleterious effects of some of these genetic mutations. Of course, we also get additional lab tests, biochemistry, and transcriptomic and various omic data in addition to the genomic itself, and as well as social determinants of health. How does lower economic status, where we live, what we eat, and how we work, those types of social determinants, ethnicity, race, and so on, and income levels, how does that affect health? So a complete electronic health record needs to go way beyond. Of course, genome has not been a part of our health record until very recently, and even now, probably less than 1% of the world's population have any genetic data about them yet. So there's a huge opportunity to help improve the human health by combining these multitude of complex data types. A lot of people wear Fitbits, Apple Watch, and so on. So you have the ability to measure your heart rate on a continuous basis. how do we use that type of data to detect atrial fibrillation, for example. That was a 500,000 people clinical study that was conducted by Apple with Johnson & Johnson to identify, only based on the patterns of heart rate, atrial fibrillation. So this is what I came to the realization just about the right time when I moved out of the National Institutes of Health lab, which I really loved, being part of a highly scientifically stimulating environment at NIH, and that's part of my journey and how I don't mean to say evolution in the sense that I am still a software engineer, so it's not like I have stopped being a software engineer. I am still a bioinformatician and a genomics data scientist. I still look at data. I have 21 summer interns working on various projects, and some projects do involve genomics and transcriptomics even today. And I do all these other hats as well. But I, instead of being boxed into one thing like being a software engineer, I enjoy the diversity and the impact that the work can have when you combine your computational and software skills with how it can impact human health. Excuse me. So switching gears a little bit, what is this? Who is a social entrepreneur? A social entrepreneur, unlike a regular entrepreneur, is different in the sense that the social entrepreneur is driven by a social problem or a single problem that can have a social impact and is driven to solve that problem, whereas an entrepreneur is primarily focused on delivering value to their shareholders or investors and customers. So for me, the biggest problem that I stumbled upon a few years ago is that the cost of bringing a single drug, there are thousands of drugs in the pharmacy, right? We all go and buy them. We take them for granted. And many new drugs are being added to the formulary, as we call it. But every single drug that gets through the basic research, discovery, preclinical analysis, and getting through the human clinical trials of phase 1, 2, and 3 to demonstrate safety and efficacy of each of these drugs, any successful drug that comes all the way through this valley of death and gets a FDA approval would have cost an average of $2.5 billion, just one drug. And it would have taken anywhere from 10 to 50 years on an average. Why is that the case? Because 9 out of 10 drug candidates that make it this far into the human clinical trial phase 1, only 12% of them get approved, 88% failure rate. And so that's what causes huge costs and timelines. And I thought maybe the cost was much higher and it's getting better and better because of technology. And I was surprised that it's actually getting worse. And so this is the problem that I have been focused on solving for the last 4 years now. Of course, I can spend a few lifetimes trying to solve this, and there are thousands of other people smarter than me probably also trying to solve the same problem. And so, but this is a problem I find worthwhile trying to solve, even if I am not successful or successful to a very small extent. It is still a worthwhile problem to solve because we need a lot of medicines for thousands of diseases which have zero treatment even today, believe it or not, including coronavirus. So what I, as the social entrepreneur in me, started with identifying what are the current problems. And what we heard, I have been presenting at conferences, listening to others, talking to customers, and stakeholders in life sciences, and I hear things like every clinical trial feels like the first ever trial undertaken by mankind. And my question to them was, why is it costing 2.5 billion, and why is it taking 10 to 15 years? What is the single problem? Right? I understand there is no magic bullet, there is no shortcut. You have to go through the process and show safety and efficacy, but why is it not getting better and better? And this was one of the answers, right? The other answer is patient recruitment is the biggest problem every single time. You know, 80% of all clinical trial participants are Caucasian historically, or at least 70% are Caucasian or white, and majority of them are males. So there is a significant lack of diversity in the clinical trial participants in terms of women, children, old age people, race, ethnicity, habits, eating habits, other types of factors. So if you are not testing the drug candidate in a diverse population, it's unlikely to be effective in the general population. It may work well for males, for example. It's a very well known fact that cardiovascular drugs work much better in males than in females, the same drug. Reason being, most of the clinical trial participants were males in those cardiovascular studies, not females. And so they were left out and so they don't get the same benefit that men get. And finally, when I look at the patient perspective, they are like, yeah, we have all these fancy tools and technologies and gadgets, but our burden still remains high because every single clinical trial visit needs to be in person. We have to travel, even though we are sick, to the clinic. And that travel can be quite burdensome. So some very specific problems that I identified in the rare diseases continuum is the challenges associated with diagnosis. So about every patient with a rare disease, not a general common condition, but a rare genetic disease, waits about 7 years on an average to get diagnosed about what disease they have. Without knowing the diagnosis or name of the disease, we have no way of going any further. It's not like a flu or an infection where you have some sort of a throat infection or a bacterial infection. The doctor prescribes your antibiotics without knowing exactly what bacteria it was. It's a suspected bacterial infection. Okay, go ahead and get a antibiotic course, right? Whereas for genetics, it's not like that. You have to perform specific tests, and it's not practical to sequence the whole genome for everybody. It's very expensive and doesn't make sense, but there are areas where it may make sense, where it's a debilitating disease and you have a family with affected and unaffected members, and you should be able to sequence at least the exome, if not the whole genome of those family members and get them to a diagnosis. Whatever it is, given all the best of the best technologies that exist even in India and US, the same Illumina sequencing platform is available everywhere now. However, the time to diagnosis is still about 5 to 7 years. And that's where the whole scientific community and the biomedical research community in the rare diseases is currently focused on is speeding up diagnosis. Within 1 year of being brought to the medical system's attention, a patient should receive their diagnosis within 1 year. Many initiatives are ongoing in that space. The second is diagnosed for a disease with an already existing approved treatment. So this happens all the time in the case of common diseases, and just because there is a treatment available does not mean that it will actually help cure the disease. It may help alleviate some of the symptoms of the disease. And so more research and more treatment development efforts will be ongoing for that disease area. But at least these patients fall in a bucket where they have some hope and some remedy there. The other bucket is patients diagnosed with a disease without any FDA approved treatments currently available. Where do they go, right? So that's where the rare disease folks, a lot of them fall in. Once they get a diagnosis, they fall into this bucket. They have a diagnosis but no treatment, and so they need to look at, are there any of these treatments going through this pipeline that I showed you, right? This valley of death. If there are treatments for a rare disease, somewhere in phase 1 or preclinical or phase 2, you want to speed up that process and get them through this valley as quickly as possible, and keep it alive, and get that FDA approval. And sometimes that you don't want to wait until it completely gets through this pipeline. So you want to get access to those treatments much faster, because the best case scenario is you don't take anything and doctors have told you that the child is only going to live for 1 year or few months. So you have nothing to lose in trying for a potentially uh, uh, useful treatment for a condition for which no treatment exists. And the biggest hope right now is our gene therapies, which offer uh, reversing the genetic mutation for the cystic fibrosis kind of diseases, where you know exactly which gene is causing the disease, and it has a very high penetrance, meaning anybody with that mutation will almost certainly have the disease. It makes sense to aim for a gene therapy, and it's a very um, rapidly growing area. You have about 1,000 plus clinical trials that are ongoing for gene therapies for several hundred rare diseases right now. And that's the type of clinical trial the patient should be aiming to get into. And finally, clinical trial should become a care option. For patients with no FDA approved treatment, you can't say, hey, I'm sorry, there is no treatment, we can't help you. Even when there is no treatment, there is potentially a clinical trial. And if there is a clinical trial, they should consider that. And one biggest myth in everywhere, including India, is that the patients who participate in a clinical trial are guinea pigs, and so they are being tested on a experimental drug, which may be completely unsafe. That's not true. These are taken extremely seriously by regulatory authorities, whether it is FDA or European Medicine Agency or the Indian CDSCO. Uh, uh, they allow clinical trials only when it is better option compared to what's the standard of care is as of today. And so it's almost always a good idea to be part of a clinical trial than not be part of any medicine or clinical trial. And if you look at the global disconnect, there's another problem that I'm looking at is, most of the clinical trials happen in the United States and European Union, and most of the rest of the world combined will account for about 10% or 20% of all clinical trials that happen globally. And if you look at India, certainly it's towards the bottom of the list of where clinical research happens. And so that's one of the areas where, of course, the population in the Indian subcontinent is about quarter of the world's population. And these historically, most of the drugs that have been discovered and developed have been coming from the Western world. And so that's a huge gap that needs to be filled. And that's why I founded the Indo-US Organization for Rare Diseases, to build bridges between United States and India to fill that gap. The third major problem which our customers and patients told us is the travel burden in clinical research, right? So in a typical clinical trial in phase 3 could run about 3 years, and every year the patients may need to travel to the clinic to participate in the clinical trial about once a month. And so that's quite a bit of travel, and not all patients are living close to these clinical trial sites, which are far and few distributed. So many of these are global, but still having a site in Boston means I have, someone in Virginia would have to take a fly to Boston just for that one clinical trial visit, maybe once a month. And that's not happening, not a surprise, because 90% of these clinical trial participants live within 50-mile radius of where the clinical trial is happening. And so unless it's close by to where they live, they don't get to participate in these trials because it's a showstopper. And so that's another problem which can be addressed using technology. And that's why I founded Jeeva Informatics Solutions to address the travel burden problem in clinical trials. How we are solving this problem is by taking stuff that's done usually as part of a clinical trial in a brick and mortar hospital site to where the patients are, performing informed consent, patient reported outcomes, video conferencing, chat, SMS, and adverse event reporting. Take them as much as possible to a mobile device that the patients may have, with whatever device they may have, and enable them to participate remotely. And there are also ways to enable home visiting nurses to collect samples, and shipping medicinal products to patients' home, and have wearable devices to collect certain data like heart rate and pulse and so on. So that's how our solution can enable 50 to 80% reduced travel, and in some cases 100% virtual, where patient would never have to go to the site. They can entirely participate on a survey-based study from their comfort of their home. So the solution involves the features like eligibility screening, electronic consent, patient-reported outcomes, video conferencing visits instead of in-person visits, and ability for physicians to make clinical assessments via a remote visit instead of in-person visit, so on. So this is typically the players involved in a clinical trial, and what Jeeva develops is a software as a service platform that is based on cloud and enables mobile bring your own device model for participants, and data is aggregated in real time, and all the clinical trial data needs to be very safely and securely stored. This is a highly regulated market. FDA can audit anytime with the sponsors, and sponsor is responsible. Sponsor is usually a pharma or a biotech company developing the drug, and they sponsor this clinical trial. And so they are responsible to ensure the security of the patient data, privacy, and all that. And so our software provides for those features. So with that, I'll come to this is my final slide, and happy to take any questions. But the journey in rare disease social entrepreneurship that began for me personally since 2012 has led me to found a couple nonprofit organizations, the National Organization for Rare Diseases India, which I led the publication of an review article of the challenges and opportunities for rare diseases in India. You can email me and I'll be happy to share all the published articles. If you like to read any of them, please write me an email and I'll share that with you. This was published in Genetics Research in 2014, as well as Indo-US Rare is another organization I founded to address the social problem of it, right? So not technology cannot solve all the problems by itself. Technology is part of the solution, but not the solution. And so you cannot, artificial intelligence cannot be a replacement for human intelligence. And so we need to remember who we are trying to help are the patients. And so raising awareness about rare diseases, clinical trials, and genetics to some degree among common public, and educating even the doctors and the researchers about the various opportunities that exist, and policymakers, to have national public health policy, state level policies, funding and budget for research and development for these diseases. So you might think when I say rare disease, they are really rare. Individual disease is rare, but when you combine, there are more than 7 or 8,000 rare genetic diseases. When you combine all of them, there are more than 300 million people affected by rare diseases globally. And in United States, that number is about 30 million. In India, it's more than 70 million. So our efforts have led to getting a definition for a rare disease in India, which any disease that affects fewer than 5 lakh people in India is considered a rare disease. And so, but that's for one disease, right? But when you combine all diseases, there's more than 70 million people or 7 crore people in India. And so it's a huge public health issue, and needs to receive the type of attention and funding from various stakeholders. And then taking it through the valley of death, through what we find in the bench, to translating that into the clinic and accelerating the drug development. Then this is a cycle and a feedback loop. So you learn from that process and you address one disease at a time, one drug at a time, and so on. So Dr. Sumalata, do you want me to share my reflections on what student level factors and faculty level factors might affect engineering education?
Yes, definitely, Dr. Harsha, we would like to.
Sure. So I just put together some factors, and not a lot of effort went into this, but I will be happy to discuss and share additional thoughts. But at least from 20 years ago, things may have dramatically changed over the last 20 years, and I do recognize some of those changes already. But certainly when I was a student in Bachelor of Engineering program, the student level factors included student attitudes, not having enough seriousness. We were not paying a very high fee. Education was affordable back then. And so, when you're getting something at a low cost, you are not necessarily as motivated and inspired to make the most of it. And of course, in India, most of the engineering education is paid by parents. For most part, some students do have to work hard to pay for themselves, and so the pain of earning that education was not as high, and many of us did not come with that kind of a commitment to really learn and understand the concepts and become good engineers necessarily. But we went through that process. Many of us, due to family and social pressures, and others because they were really passionate about what they wanted to do. They had clarity at the time. As we all know, the social pressures are pretty high in India around, if you're not an engineer or a doctor, you're no good. I don't know if it's changed these days, but back then it was like that. And within engineering, we commonly joke in social circles here, after 20 years, that one of my friends in civil engineering said, when he went to weddings and social gatherings, and someone asked, what are you doing, and he says engineering, the very next question is, which branch? And when he says civil engineering, oh, civil, so it's like civil is no good, right? So that type of belittling of disciplines and education and so on in the social circles puts a lot of pressure on students, and that's an important factor to address. The third is exposure of the students to what the opportunities are and what the application areas are. That's significantly lacking. One, because there are no postgraduate level like MS and PhD education happening at the same institution. For example, when I was at Dr. EIT, I do believe there was a master's program, but we rarely ever interacted with the master's level students and our faculty members and any industry players that would come. So limited on-campus recruitment and so on. And also collaborations between academia and industry was not as much. And so the students are more worried about, because of social pressures and family pressures, which branch of engineering would earn me the highest salary after graduating, right? So that becomes a big criteria. And then I already spoke about the cost of engineering. So I would like to hear how these factors have evolved over the years, but I thought those played a big role back when I was going through college. At the institution level, back then, after I graduated my bachelor's degree, I was called upon by the head of department in computer science to provide as a temporary faculty to help fill for lack of qualified instructors and faculty members in engineering college. And I was just finished bachelor's degree, and I was teaching seniors in our fourth year engineering computer networks. I was teaching soon after I graduated. That was just for one semester, and I know for sure that things have changed in that regard in terms of the university-wide requirement for at least a master's level degree now for faculty members, and have PhD either in progress or on schedule to complete within a certain period of time for them to be qualified as in faculty, which is a big step forward in the right direction. And the quality of classroom teaching, of course, back then, because of the lack of qualified faculty members would suffer. Some subjects had really good qualified faculty, whereas others suffered from lack of it. Practical exposure of application areas. Like, little did I imagine when I was going through my Bachelor of Engineering that I would end up in genomics, precision medicine, and I would talk so much biology and medicine today. I had no clue that was going to happen. But gaining exposure to this webinar series is a great way to provide this kind of exposure to current students, so they have an idea and understanding of the various directions in which they can take their knowledge and learning. And it doesn't have to be an IT company. Computational science is now part of all disciplines and all industries, verticals. And then, not having higher education in the same institution, like a PhD program and research projects and funding is a big factor, I think, that can affect the quality of engineering graduates. So with that, I conclude my talk, and thank you for this opportunity and listening. Happy to answer any questions.
Thanks, Dr. Harsha, for the brief session on genome engineering. Though it is a very vast area, you have given a little bit glimpse on what actually it is. We have a couple of questions. I would like to show that. The first question is, how was the reference genome chosen in genome sequencing?
Very good question. So the first genome sequenced was of I believe either Watson or Crick, the two scientists that discovered the double helical structure of the DNA, Watson and Crick model. So one of their genome became the reference genome. They had to pick someone, and both of them had aged and and I think fairly healthy. And so they had to pick. Um, but it's a very good question in the sense that who is the normal human being, right? Like if someone else is, if I am normal, does that mean everyone else with relation to me would be abnormal? And it's not a single reference genome in theory, right? Ideally, you want to have thousands or millions of genomes that could collectively become the reference, and that's the type of thing that's happening now at population scale, now that large amounts of data is available. But um, to begin with, and for getting quick processing and even fairly accurate processing, uh, the first reference genome was, I believe, of either Watson or Crick.
Okay. Over to the second question. What is the effect if this reference genome is changed?
It's the sequence has remained the same or fairly constant. What's changing is the annotations. So, if you have a genome, of the 23 chromosomes, right? And, each chromosome can be tens of million or 100 plus million in length in terms of the DNA sequence. And you need to identify genes. So there are gene prediction algorithms. And once you identify the genes, you need to annotate all the introns, exons, where is the start of the gene, where exactly is the end of the gene, and the promoter regions where how the gene is regulated, meaning how the gene gets primed for transcription into mRNA. So those types of detail are what we call annotations, and those annotations are the ones that have been evolving and refining over the years. But the sequence itself has been fairly stable.
Okay. The next question is, does genome data help to predict the possible diseases a person can get in his lifetime so that precaution can be taken well in advance?
Yes. So preventive medicine is not something that's happening as much as it should. And there's not as enough resources and attention going into preventing genetic diseases as there is to treat the genetic disease. And so that's a very important area. I think prevention is always better than cure. And we need to look for ways in which prenatal genetic testing and premarital genetic testing between the couples. So it's one thing that, certainly for the Indian communities which marry internally within the families, uncles and aunts, kids, and so on. So that has to be absolutely stopped, and that awareness needs to come, and that's one way we can definitely prevent the genetic diseases. And that education can occur at various levels, at a mass public level, but also in premarital counseling as well as prenatal counseling. During pregnancy, also certain known genetic mutations can be identified from the mother's blood, even in the first trimester, with the prenatal screening techniques that are now available. And so that's becoming more and more widespread, but more awareness is needed there, and prevention is definitely possible, and there are methods to do that, both genetic as well as education level.
So just on curious part, any software or tools are they available for the prediction of such data?
Yeah. Software tools are all available. We need to generate the data first for the say the couple, for example, and there are services available. Like in the US, we have 23andMe. In India, there is a company. I'm blanking on the name, but they have something called Genome Patri.
Okay.
Just like Janam Patri, they make Genome Patri. For every baby that's born, they can pay and get that service, and they get their the baby's genome. And also they have uh, uh, marriage patri kind of a offering as well, so where the couple can get counseling on their genetics before planning their family. So those types of consumer level services are already becoming available, and they are available even in India now.
Fine. Okay. So next question is, will data keep changing for a person based on his lifestyle, the genome data?
Good question. No, the genome in the cell of every cell of the body is fairly stable and static throughout the life. What changes is their epigenetics, and that's based on the environmental factors. Like UV radiation can cause a mutation in some one or more cells that are exposed. People going and sleeping on the beach in hot sun might get some UV light to cause some random mutations, but the actual genome itself is does not change, and it's static. Exception is cancer, where the cancer cells have a lot of widespread genetic mutations in cancerous cells only, but not the rest of the cells.
Okay. Yeah. The last question. Can you list few of the rare diseases on which genome engineering has been successfully applied?
Yeah, there's a lot of them. I mentioned one, cystic fibrosis, and there is Rett syndrome, muscular dystrophies, spinal muscular atrophy is another rare disease. It's a neuromuscular disease which has Avexis, a gene therapy, one of the success stories recently that came about, and a rare form of blindness, a retinal disease, also has a gene therapy. So, surprisingly, even though a lot of funding goes to cancer in general for research, eye diseases have been at the forefront when it comes to gene therapy and so on. So, one of the earliest gene therapy was conducted in dog for retinitis pigmentosa, and that was on a gene called RPE. And that was only in the dogs, but even in humans, one of the 4 approved gene therapies today by FDA, and we are expecting that number to become 10 by end of this year. The first 4, one of them was for an eye disease, and another is spinal muscular atrophy, which is a neuromuscular disease.
Okay. Yeah, I think that's all we have from the audience. Okay. Thank you very much, Dr. Harsha, for giving us a note on what is a genome engineering, the different challenges which we can have, and how to predict the genome data, and how we can take a better care of ourselves by looking successfully by the predictions and all. And also thanks for listing us a few on rare diseases which are there in the genome engineering. Thank you very much, Dr. Harsha. I would like
Certainly.
to have more contact details so that if anyone who are interested in seeking their career or any research in this area can come up with the things and can approach you at any time.
Absolutely. Yes,
Yes.
certainly.
Yeah.
They can reach me at ceo@jeevatrials.com.
Okay. Yeah. Thank you. Noted.
Great. Thanks for the opportunity, and enjoyed interacting with all the audience questions. Appreciate you listening.
Yeah. Thank you. Thank you very much, Dr. Harsha. Yeah. Bye. Have a nice day.
Bye-bye.
Bye-bye. Dear participants, we will be sharing the feedback form right away. Request all of you to please share your valuable feedback to us. It will be kept alive for about 1 or 2 hours. Request you to fill the feedback form and get back to us. Thank you.